Good afternoon. Welcome to our IDDD roundtable. Today's topic is peptide drug development and delivery. Roundtable, we have two distinguished speakers. Before I introduce to the speakers' talk, I would like to go over a little bit about our center, which is a Gulf Coast Consortium Center for Comprehensive PK/PD and Formulation. This center was originally founded by CPRIT in 2018, and we are focusing mainly on the preclinical drug development, focusing mainly on the small molecules. It's a collaboration between our university, Texas Southern University, University of Houston, and the Gulf Coast Consortium. Over the past 5 years, we have provided quite a lot of services across the whole state of Texas with multi-institutions, with investigators, and we have made a pretty good impact in terms of the grant funding, as well as impact on pushing for the first-in-class and the first-in-kind novel cancer therapies. And the center was recently renewed. We got another 5 years to provide more services in the next 5 years for our center. This is a glance of our research facility at Texas Southern University, which includes bioanalysis, mainly using the LC-MS instrument lab, accurate mass spectrometer for unknown metabolite identifications. We have formulation and nanotechnology lab. We also have biopharmaceutics as well as pharmacokinetics and pharmacodynamics lab facility here on campus. We collaborate with University of Houston mainly on the more complex PK/PD modeling and simulations. And we collaborate with the Gulf Coast Consortium mainly to provide networking and conferences for the updated development in the area. And that's our website. Now I'm going to go to first introduce our first speaker. Our first speaker is Dr. Phillips Kesisoglou. And Dr. Phillips Kesisoglou is a distinguished scientist from Merck Drug Company, where he is currently leading the exploratory biopharmaceutics efforts across modalities in pharmaceutical sciences department. Phillips has 20 years of experience in the field of biopharmaceutics and formulation development, pharmacokinetics, PBPK and IVIV modeling as relates to the clinical and drug development and CMC regulatory applications across modalities, including oral peptide drug delivery. He has been a key contributor to several new drug applications across therapeutic areas, and he has authored over 95 publications. He is currently serving as an editor for Journal of Pharmaceutical Sciences and on the editorial advisory board for the AAPS Journal and Pharmaceutical Research. In 2017, he was elected as a fellow of American Association of Pharmaceutical Scientists. Without further ado, Dr. Kesisoglou.
Thank you, Don, for the nice introduction and for the invitation to present today. So let me pull up my slide and hopefully this will work as intended. I'm assuming everyone can see the slides right now. So
not yet.
Let me click this again.
Yeah, we can see it.
Okay, perfect. Thank you.
you.
So for the next half an hour or so, I will share some of our experiences with the clinical development of an oral macrocyclic peptide, MK0616. But to start, maybe some general comments around oral peptide delivery. So peptides are commonly used for therapeutic purposes, and there is approximately 80 approved peptides as marketed drug products, the vast majority of them being administered parenterally, either IV, IM, or subcutaneously. There is only 5 products on the market based on oral peptides for systemic oral delivery. And I mean, those 5, a couple of them are mostly closer to small molecule than an actual peptide. So there is definitely significant opportunity on the oral peptide delivery, and there is an increased emphasis in this area. If you follow the field, there is several clinical trials ongoing across different companies right now looking at the oral peptide delivery. The oral peptide delivery, however, is challenging. The intestinal epithelium and the intestinal environment is not friendly to peptides. There are barriers that are present in the gut lumen. There are enzymes responsible for breaking down many of the peptides, similar to what they do with proteins, which is part of our nutrition. Mucus across the intestine represents a secondary diffusion barrier for peptides. And finally, probably the even most difficult barrier to overcome is the intestinal epithelial barrier, where peptides are generally large molecules, and the intestinal epithelium is not designed for absorption of large compounds. So the size limitation for oral delivery has been studied, for many decades. It is very well documented. Most of the hydrophilic compounds are absorbed paracellularly through the tight junctions, and tight junctions have a size limitation to what can go through. On the left-hand side is a graph that shows the impact of the hydrodynamic radius of a molecule. In this case, they were studying PEG type of compounds. And you can see that the flux significantly drops as you increase the hydrodynamic radii of a compound. The paracellular pore size is considered roughly in the 8 to 9 Armstrong diameter, so that typically equates to a 5 to 600 Dalton molecular weight. And that is actually reflecting the bioavailability of oral compounds. On the right-hand side, there's a graph that shows the bioavailability as a function of molecular weight in rat, humans, and dogs. And you can see that pretty much when you hit the 500, 600 milligram, 500, 600 molecular weight range, the bioavailability drops significantly. Dog is a little bit higher bioavailability than humans, but since we're talking about clinical trials here, again, once you hit that high molecular weight range, we're talking about single digits bioavailability. So what's currently on the market? There is technically five peptides on the market. The one that everyone is more familiar with because it's in the news a lot lately, semaglutide, the GLP-1 compound from Novo Nordisk is given as a oral tablet with a SNAC, which is a fatty acid derivative, as a permeation enhancer, and it has a bioavailability of 0.5 to 1% or so. I think the new formulations are closer to 1%. There is a couple of other compounds on the market. Octreotide is the other, relatively larger peptide that is on the market as Mycapssa. It is also using a lipid mixture as a permeation enhancer, and that bioavailability is also 0.5 to 1%. The exception to this low bioavailability is the cyclosporine type of compounds, voclosporin and cyclosporin. these are still technically cyclic peptides. However, because of the conformation and the molecular structures and everything, these actually do show good bioavailability, especially cyclosporine availability. 10 to 90%, extremely variable, but it's formulation dependent. And the last one on the market is desmopressin, molecular weight roughly 1,000. It is on the market as oral tablets. Actually, it doesn't use any permeation enhancers or anything, but it has a bioavailability of 0.16%. So you can see that generally the bioavailability of these peptides is very, very low. I mean, we're talking about currently 1% being the target for these therapeutic peptides. So as I mentioned, enzymatic degradation is one of the concerns for oral delivery of peptides, but that can be solved through molecular design. So generally speaking, for the macrocyclic peptides that we are working on, we are not significantly concerned about enzymatic degradation in the GI lumen, at least within the context of an oral absorption time, like so several hours. The peptides are stable. So our primary worry is getting the compounds through the epithelial barrier into the systemic circulation, and that's where permeation enhancers come in play. There are permeation enhancers that you would generally see cited in the literature falling into two categories: those that focus on tight junction opening and primarily facilitate the paracellular absorption, and those that theoretically can do transmembrane fluidization and may facilitate transcellular absorption. The most common ones are fatty acid deliveries. Sodium caprate or sodium decanoate is one of the very high cited in the literature, and SNAC, which is another again, derivative of fatty acid that's in the Ribelsus formulation. And permeation enhancers have been studied for decades. I'm citing a few papers here that discuss how these can work in terms of impacting bioavailability of oral compounds. Initially, a lot of these permeation enhancers were tried in the small molecule space with the hope that it will increase permeability of BCS3 compounds. Though that hasn't really played out much, but they're becoming much more interesting in the context of large molecule delivery. I mean, we'll be talking peptides today, but there are efforts going out there on delivering even larger constructs like proteins and monoclonal antibodies. So the most common permeation enhancers are again fatty acid deliveries. For the talk I'll be giving on the MK0616, I will mention Labrasol and Caprate because these are the two ones we used. Labrasol is a mixture of PEC400, C8, and C10 fatty acid esters. So it breaks down in the GI tract to the corresponding fatty acids that act as the permeation enhancer. And it has been used in many marketed products because it's also a solubilizer. It helps with solubility, but there is no marketed product that actually uses it as a permeation enhancer. Sodium caprate, again, it's a fatty acid. It's present in milk. I mean, especially like goat milk has high amounts of sodium caprate, so it is part of diet, and it has been used in several clinical trials, but there is no marketed product yet. And then SNAC is currently used in Ribelsus. The way we study permeation enhancers prior to moving to the clinic is through cell line experiments. These are some experiments I'm showing here in KECO2 cells. We use at Merck both transwells and gut-on-a-chip type of systems. And these examples show on the left-hand side the transwell, on the right-hand side the chips. This is just the FD3, the fitsidextran permeability as a marker. You can see, sodium cuprate or sucrose and monolaurate, different permeation enhancers, clearly show you a dose-dependent increase in the permeability of the marker and corresponding drop in the TIR value, indicating that the permeability is increasing and the tight junctions are opening. We can do the same experiment with actual, peptides. On the left-hand side, like Fitzy insulin. On the right-hand side is octreotide. And again, compared to untreated, we see first an increase in permeability as you look either at transwell or the chip system for both of the peptides. We actually do see differences on the permeability as an absolute value and as a relative value, depending on what system you use. So, using a transwell will not give you the exact same result like using an oligo-on-a-chip. And it will not give you the same result as using an in vivo animal, for example. But directionally, generally, the systems tend to tell you if a permeation enhancer is actually improving the bioavailability of the peptide. I will switch gears now to discuss a little bit about MK0616 and some of our clinical experience. This is the structure of MK0616. You can see it's a macrocyclic peptide. The molecular weight is roughly 1500. The one I'm showing here is the hydrochloride side that was used in all the studies I'm going to show you. It's stable, as I mentioned, so stability is not a concern for us in the GI lumen, and it's not permeable. So intracellular permeability is extremely low. It's like, less than 1 times to the 10 to the minus 6. So for those who work with cell lines, this is like less than the permeability of mannitol. So it is clearly not going into the cells. It is highly soluble. It's soluble like several mg per ml across the GI tract. And then preclinically, it demonstrated good bioavailability, meaning like 1 to 2% in preclinical models, in monkeys, for example, where we formulated with an established permeation enhancer, and obviously it had the desired pharmacodynamic effect. So we went to the first in human study. This was the first oral peptide we're taking to the clinic. Try to answer a few questions. In any first in human study, you always need to check that the compound is well tolerated. So that's standard, that's nothing new there. We're going to see that the MK616 is getting absorbed. We had seen the evidence preclinically, but you need to see the evidence clinically that you're getting bioavailability. We were expecting high variability, given that the bioavailability is like only 1%. The common sense will say, you will have high variability, and that has been seen with the peptides on the market. We're going to see if permeation enhancer will help exposure. And finally, we want to see whether we can use sodium cuprate. The toxicology program was pretty standard for early studies. We hadn't seen really anything in terms of toxicity, so we weren't really concerned about the systemic exposures going above any specific range. And again, we were expecting low bioavailability, so that wasn't a significant concern. So first, these are the data from our first in human study, up to 300 milligram dose. The compound was well tolerated in the first human study as expected. There wasn't like anything out of the ordinary stuff you would get in a first human study. And the compound was well absorbed. You can see we have measurable plasma concentrations up to 168 hours across the entire dose range. The surprising outcome was that the variability wasn't high. The variability we're seeing for the compound is not any different from what we typically see for an oral small molecule, BCS, 2 or 4. So we're estimating the bioavailability clinically to be the order of 1 to 2%, but the low variability was, a positive outcome, a positive surprise for us. We confirmed that permeation enhancer helps with exposures. In our very early dosing, we used the Labrasol for ease of making those supplies at the site of administration. And you can see comparing the data with and without Labrasol, there is a 2 to 3-fold increase in AUC and like a roughly 4 to 5-fold, if not higher increase in Cmax. So permeation enhancer definitely helped increase the bioavailability of the compound. But the compound is also clearly absorbed on its own. So despite its molecular weight, there is inherent absorption of this macrocyclic peptide. We then wanted to check, if we can use sodium caprate over Labrasol. We were more interested in the sodium caprate because, ideally, we're interested to make a solid dosage form. You can make a tablet using sodium caprate, while Labrasol is a liquid system. So we basically compared those two at several panels. I'm just showing you here the comparison at 100 milligram, and they were pretty much indistinguishable. I mean, within the noise of the first human study, these look pretty much identical. We actually dosed in the study anywhere from 360 milligram to 1800 milligrams of sodium caprate. As you're increasing doses, it's just more units, so the total amount increases naturally. But we didn't actually see a significant impact in bioavailability, going to higher caprate numbers. Even the 1800 milligram as a number is still lower than what you would typically have from dietary intake. So our concern wasn't really concerns about the sodium caprate having any, AEs or like negative effects. It was more, can we keep the amount of sodium caprate low so that it makes like a more manageable tablet for future development. Going in the study, we were expecting to see a negative food effect. Most of the peptides show negative food effects. Some of them show actually zero exposure with food. Some of them have like, a significant reduction. In our case, we saw at like a roughly 3 to 4-fold reduction in exposure when the peptide is taken after the meal. But if you take the peptide, wait half an hour to eat, and then you take your meal, then that absorption has happened already. So there is no negative effect of to the peptide anymore. So the data, I mean, this is how the marketed peptides, like the Ribelsus and octreotide, are also administered. They all have this half an hour wait period between taking your drug and getting a meal. They're all using an overnight fast. I think some of them saw absolutely no exposure, with food. In this case, we had some exposures with food, but still it's clearly a significant effect. The next step will be, okay, this shows all the PK impact, but does it work? Does it do what it's supposed to do in terms of its LDL reduction? And these are like some of our early data from our early studies where we saw robust LDL reductions even at 10 milligram doses, which is like a relatively low dose. What was interesting in the study in terms of understanding the absorption of the compound, you can see that even when we went down to 180 milligrams cap rate, we still had a good LDL response. So you don't need much of the permeation enhancer, at least for this compound, for it to be absorbed. And when we give it with food, you clearly lose bioavailability, and that directly corresponds to a reduction of your LDL impact. So, like, you lose half of the exposure, and then your 10 milligram behaves closer to a 5 milligram in terms of its like pharmacodynamic effect. These are all good. These are all controlled studies, phase 1 studies. They are very controlled because they happen on site. The question was, would this work in a larger, a bit less controlled situation? So we did run a phase 2 study. The results have been published. You can find much more details on what I'm sharing here today. We dosed, between 6 and 30 milligrams of the compound, and we saw robust LDL reductions across the doses. There is clearly a dose response, although you can argue that at some point, the response is pretty flat once you had this, like maybe somewhere between 18 and 30 milligrams. But it's clear that we're getting the bioavailability we want, even in a setting of phase 2 studies. So overall, very positive for the compound. In terms of understanding the biopharm and the absorption, the other thing that came up during development a little bit more recently was around the administration restrictions. If you look at some of the marketed peptides, they have reported that the amount of water you take impacts the bioavailability. So the concept is that when you take a larger amount of water, you dilute the permeation enhancer and therefore there's less concentration of the permeation enhancer, and it reduces bioavailability. So we didn't think we'll see an effect because we had seen pretty consistent PK across multiple dosing scenarios. But we did run a dedicated study where we dosed the MK0616 with 240 mils, 120 mils, or 50 mils of water, and there is really no impact on the bioavailability. These are like relatively, these are just the relative exposures, AUC and Cmax, relative to the reference, which is the standard 240 mils water that's used in clinical studies, and really there is no effect. So this compound specifically is not affected by this water volume effect. It will be compound dependent. We've seen other compounds, again, marketed compounds clinically, or we've done preclinical studies on other compounds that have shown some effects. In terms of biopharm research, there's a lot of interest recently in expanding physiologically based PK models to the oral absorption space. You might have heard the terms physiologically based biopharmaceutics modeling. But all these models have been really developed with small molecules in mind. So the question is, can a PBPK model really handle an extremely low permeability and a permeation enhancer effect? And the answer is not yet. We're still missing the ability to mechanistically incorporate permeation enhancer effects in the models. I'm not going to go through the details of each of the simulation. This is just the same data fitted with different assumptions on how the compound gets absorbed, whether it's absorbed transcellularly, paracellularly, or transcellularly, but with like fast movement out of the enterocyte. And as I always say, I have never seen someone who cannot fit data in a model. Like all the models always fit the data. The question is any of this actually physiologically relevant, and which one does it actually reflect reality? And if you do the next step and you start taking a closer look to what the model is telling you, you can start saying that yes, all of them fit the data, but for example, this one tells you that your peptide gets stuck in the enterocyte for several hours. This one goes through the paracellular junctions. So it doesn't get stuck in the enterocyte, but then it's still pretty slow absorbing. So which one of these is true requires many more additional data to validate the model. So I'm hoping as we're seeing more and more peptides make it to the clinic orally, we'll start seeing also these models advancing to more sophisticated solutions. So some general questions for biopharm research for those who are you studying in this field. I have started wondering, since looking at this peptide and also other peptides we have in the portfolio, have we historically overestimated the oral absorption variability, or at least the impact of permeability on permeability, or bioavailability? Again, conventional wisdom will tell you low absorption results in high variability. But I mean, we didn't see this. I mean, the variability here is normal. It isn't any different from a typical oral small molecule. When we're talking about permeation enhancers, is there a minimum amount of permeation enhancer required for effect? And is that conserved across peptides? If you look at most of the literature for caprate, it will cite 500 milligrams as the effective dose. However, we saw positive results in lower doses. The next step is like, how can we translate this from clinical to preclinical screening? So when the next peptides comes in, we can do this screening effectively. And as I mentioned, PBPK models, I mean, I think there's a lot of interest in the area. I'm personally more interested in them to answer clinical questions. For example, there's a lot of interest from agencies on whether permeation enhancers will create drug-drug interactions with other compounds. We've done plenty of studies. We don't see much, but hopefully, as we are moving in this field further, these models will continue advancing, and hopefully some of you who are working in this field will work in these areas. So with that, I will conclude my talk. I think I'm about on time. And I just want to thank several of my collaborators and the 616 team development team members. My email is first name, last name @merck.com if there's any follow-up questions, and you can also find me on LinkedIn. With that, I'd like to thank you for your attention and calling in today, and I'll pass it over to Michael and the next talk.
I think Dr. Leon somehow, yeah, so Michael, please continue.
Sure. Let me start by thanking the organizers for letting me come here and speak today. The opportunity to talk to people who think about peptide drugs was too good to pass up. By way of introduction, my name is Mike Lorenz. I am the chair of the Department of Microbiology and Molecular Genetics at the UT McGovern Medical School. What I am going to share with you today is the story of a project that at times has appeared too good to be true, but represents a 50-50 collaboration with the person you see on the screen here today, Dr. Danielle Garson, who is also in my department at McGovern Medical School. So I'm a mycologist, I'm a fungal biologist, and what I'm going to do today is to lay out the problem, and that is the substantial need for additional antifungal agents to use clinically. And I'm going to give you some stats at the beginning that I think will probably shock you, unless you think about fungal infections more than most people do. And then going to describe to you a project that Danielle and I began to look at interkingdom polymicrobial interactions that has generated this potential antifungal agent. And we'll walk through some animal data and some optimization, and finally a little bit of mechanism of action studies. Okay, so the problem of fungal infections. This was published by several colleagues of mine more than a decade ago, but the numbers, if anything, have only gotten worse. I'll show you one example of that here. These are the number of life-threatening infections by, at the top of the screen, the big 5 fungal pathogens, and their mortality rate. And if you do the math on this, which they did for us, there is an estimate of over 2 million life-threatening fungal infections and an upper limit of deaths at 1.5 million. This exceeds the worldwide death toll from malaria, tuberculosis, or HIV. So this is a problem, and it's one that has only increased in significance over the last several decades. David Denning, who's at the University of Manchester, is a clinician scientist who dabbles in epidemiology and aspergillosis, 2 things that are not heard in one sentence very often together. Excuse me. 2 years ago, he published an updated estimate of this, that there might be 6.5 million invasive fungal infections and 2.5 million attributable deaths. So these are enormous numbers, and ones that I think people who do not work in the clinics, do not work on ID wards, don't appreciate how big a problem this really is. It's big enough a problem that just a couple of years ago, 2 or 3 years ago, the World Health Organization issued for the first time in history a fungal priority pathogens group. You will see in this talk a little bit of data on Cryptococcus neoformans, a little bit of data on Candida auris, and a fair bit of data on Candida albicans. So we're working here in the most problematic pathogens. So why is this such a big problem? Well, this is the timeline for antifungal development over the last half century or more. And what you see here is there are not very many lines that point to this timeline. Our first antifungal was, our first antifungal option was identified in the late 1950s. It is a problematic drug because it's quite toxic. We did not discover the first azole antifungal, which is the front line in most cases, until late 1960s. It took a dozen years to get that to market. The third category of antifungal drugs, the echinocandins, which by the way are lipopeptides, they were discovered in 1973 and made it to the market in a whirlwind only 28 years later. So we have issues here. And part of the issues are that developing antifungal drugs is very, very difficult because the cell biology of a fungal cell and the cell biology of a human host cell are virtually identical. A colleague of mine, Ambro Van Hoof, down the hall from me, did a little bioinformatics and identified all of the fungal genes that are essential in fungi and are not found in mammals. And that number was 6. That's right, 6 genes, and 2 of them are already targets of these antifungal agents, of approved antifungal agents. So this is hard, and we were searching for different ways to address this problem and to think about whether we can identify new pools to fish in, essentially looking for antifungals. Now, the typical drug company screen of find a million compounds and screen them against fungi, I think that's played out. I don't think we're going to find another antifungal drug in that manner. So we need to be a little bit more clever. And our approach was to think about Candida in its natural environment. And its natural environment is us. So it lives in our gastrointestinal tracts, oral cavity, on the skin, the genitourinary tract. So it's part of the microbiota, and it is probably the most abundant fungal component. Not probably, it is the most abundant fungal component at most of those sites. In those sites, it has interactions with a variety of bacterial co-travelers here. And I used to have on this slide that there were synergistic interactions and antagonistic interactions between these. But as it turns out, the line blurs very much between what is synergistic and what is antagonistic. Pseudomonas outright kills Candida albicans, but these are co-isolated in, say, ventilator-associated pneumonia and the CF lung quite frequently. Staph aureus and Candida appear to make better biofilms that are more drug resistant than either species alone. And this can go on and on. So Danielle and I were thinking about what other bacteria we might be able to think about. And we came across this paper, which this slide is busy, and I don't expect you to read anything but the punchline. And that is these investigators looked at almost 70,000 clinical samples from 2 decades in their hospital and identified a little over 5,000 that had Candida in them. And of those 5,000, 22% of them also had Enterococcus faecalis. This was 3 times higher than any other bacterial passenger on this list. So what is Enterococcus? It's a gram-positive bacteria, and Enterococcus and Candida albicans live in the same places in the body. They infect the same kind of patients, the same risk factors. They are both equally difficult to treat because they are drug resistant and recalcitrant to most of our current therapies. So we thought that this was something interesting. It didn't hurt that Danielle is an Enterococcus expert, and I pride myself on knowing a little bit of something about Candida. Danielle's lab has long used a simple nematode infection model system in which C. elegans is fed a variety of bacterial pathogens, and they die or they don't die. And mammalian virulence factors have been identified through this model. So somehow this little simple roundworm is actually a really effective model for human infection or mammalian infection. So what does this look like? So in the middle of this image is Enterococcus infected worms. You just put them in a dish. The C. elegans eats the bacteria, and then the Enterococcus causes a breakdown in the normal architecture of the gastrointestinal tract. You can see here in this close-up, there are attaching and facing lesions here. You don't see the other side of the intestinal tract. The architecture of the brush border of the intestine looks just like it does in us. So that's one reason why this is a good model. You see none of that when they're infected with Candida, the dark cells here, both yeast and these longer extended hyphae. There is no architecture, there is no gut. Candida has essentially liquefied the inside of this worm, and they die quite quickly. To our surprise, bordering on shock, when we put them both in together, the gut architecture of the worm is perfectly normal. The larger cells are Candida, the smaller cells are Enterococcus. You can see the brush borders here, they look absolutely normal. There are no fungal extended hyphae in this situation. And the worms live as long as they do on control media, which is feeding on E. coli. Now, this was an observation by Melissa Cruz and Carrie Graham, working between our 2 labs. And Carrie then discovered that we didn't need Enterococcus for this. We could simply take spent culture superdate. So whatever Enterococcus was doing to Candida, it was secreted in the media, and that alone was sufficient. We told Carrie to get in the cold room and start purifying things. And neither Danielle and I are biochemists, and Carrie wasn't a very good biochemist either. But she came to us one day and said, I know what it is. And she had trolled through, as a second-year PhD student, trolled through transcriptomic data and said it has to be this gene called EF1097. Our response was like, okay, Carrie, I mean, you can pursue that, but you can't guess this. And it turns out that she did guess it, and she was right. EF1097, we now call INT-V. It is a bacteriocin with some activity against gram-positive bacteria. It's not a pore former, but it does kill these bacteria through some mechanism. It is secreted as 170 amino acid pre-propeptide that is cleaved on the outside to get rid of the signal sequence to 136 amino acid propeptide, and then down to a mature form on the outside of the enterococcal cell through a proteolytic cleavage event and the introduction of a disulfide bond that is required for its activity. This mature version is 68 amino acids, 7.2 kilodaltons. When we synthesize this peptide, this is again data from the worm model. You can see E. coli in the red line. They live out to about 20 days. If you infect them, if you let them feed on Candida albicans, that lifespan is cut to 10 to 14 days. And if you give the synthetically produced NV at 0.1 nanomolar, lifespan is completely normal. So it works in the worm model. Does it work in a mammalian model, which is, of course, what we really care about? And so we used a model of oropharyngeal candidiasis oral thrush. This is common in AIDS patients and infants and people with dental implants and other things. And what you see here are animals infected with, orally infected with Candida albicans and treated with a vehicle or with our synthetic peptide. I think you can see clearly here, there are a few fungi here and here. They are mostly in the less pathogenic yeast form. Their association with the epithelium is fairly peripheral. In contrast, the untreated animals or control-treated animals, you see a huge amount of neutrophil infiltration, inflammation, tissue damage as these, again, magenta fungal hyphae are penetrating through these tissues. We quantitate that data. We do this by qPCR or fungal DNA. And you can see here on day 3 after treatment and day 5, this is actually a 27-fold difference in fungal burden between our treated and untreated samples. Now, that was good enough for us to think that this was something we ought to pursue. And I'm going to make a comment for any of the trainees, students, postdocs, even junior faculty. Virtual presentations like this are nice. It lets you get your message out to a wider group of people, but it's no substitute for going to a meeting. And so Danielle presented this data at a molecular pathogenesis meeting a few years ago. And these two people, Alexa and Carla, were in the audience. And they run an NIAID-funded Center for the Structural Biology of Proteins Relevant to Infectious Disease. And we had a little bit of like a blind date kind of thing at a coffee break after that, where they were saying, are you interested? And we said, are you interested? And in the end, Alexi and Carla hooked us up with Peter Stogios at the University of Toronto. This is the crystal structure of the 136 amino acid propeptide. We couldn't make the 68 amino acid version because it's toxic in E. coli, which already tells you something there. But you can notice in this structure, there are 6 alpha helices that are caging a 7th alpha helix, which is in red. And when Peter showed us this data, Danielle and I looked at each other and we go, it can't be that easy, can it? And in fact, it is that easy. So if this is the structure, the helices that are in the beige color are the ones cleaved off, and that has the effect of releasing one face of this red helix to the solvent. So we asked a question, well, could we just make that? And that's not going to work, is it? And it worked. So peptides derived from that red helix down to 12 amino acids are fully active in the nematode model, which again was a surprise to us, but sort of fit the way this project has worked since the beginning. This is what the 12 amino acid peptide looks like. I think you will recognize, for those of you who know something about antimicrobial peptides, this looks nothing like a conventional antimicrobial peptide. It's quite hydrophobic. It has a couple of hydrophilic residues. I will show you in a minute. They're not required for activity. And so 12 amino acids, that's all we need. Is this then active in mouse models? This is a prophylactic model, meaning we start treating on day 0 of oropharyngeal candidiasis. And you can see our short 12-mer peptide works just as well as the longer native 68 amino acid peptide. I said that's a prophylactic model, and that's what we used for a long time, but it also works in a therapeutic sense. So these are untreated animals at day 3 and day 5. At day 3, we sack these animals to get a baseline, and then another cohort we started treating at day 3 and compared them to treatment prophylactically from day 0. And you can see we go from this fungal burden to the green dots in a 2-day period. So this works in a therapeutic sense as well. Now, these animals are simply getting this in their drinking water, so there's no fancy preparation here. It's at the drinking water at 100 nanomolar concentration. The mice drink whatever they will in a day. Another problem with fungal infections, particularly Candida infections, is that of intravenous catheters. These are seeded with Candida organisms, which create biofilms there. We turned for this model to our friend and colleague, David Andes at the University of Wisconsin. And you can see these are untreated catheters with this fuzzy, by EM, this fuzziness in the catheter. These are sectioned transversely with thick, very complex fungal biofilms. If we treat with 100 nanomolar NV, this is the 68 mer, then it completely sterilizes the catheter. Dave says he's never seen that with anything he's tried before, including clinically approved antifungal drugs. When we got down to our 12 amino acid peptide, we got a little greedy and we took it down to 20 nanomolar. And now we don't have quite the same reduction, but it still works quite well. And you can see the difference between this biofilm and the treated one. So that's another very relevant. In this case, it's a rat model for infection in the catheter. It may have some potential for broad spectrum activity. It goes back to our C. elegans model system, but it is effective in this model against the emergent drug-resistant pathogen Candida auris. It is also effective against azole-sensitive and azole-resistant isolates of Candida albicans, and it is effective against Cryptococcus gattii, which is an organism. It's a Basidiomycete and about 500 million years removed from Candida albicans. So that gives us some perspective that what we're seeing here is not a niche product with Candida albicans, but might have more broad specificity. Now, I showed you the 12 amino acid peptide worked. Shorter peptides of 11 and 10 amino acids work, but they're not as effective. Sean Guha was a postdoc in Danielle's lab who had great experience in peptide biochemistry. And so he wanted to see if we could take this 10 amino acid version and make it better. And the first thing he did was an alanine scan of this peptide. And what you can see here on the left are the substitutions that he made. Alanines, all these locations, including the hydrophilic glutamines here. And none of these broke the peptide. Every single one of these worked. And so we went about trying to see if we could make this peptide better. To do so, Sean synthesized about 2,200 peptide derivatives of the 10 mer and the 11 mer and tested them in 2 orthogonal models. One was infection of C. elegans, and one was in vitro biomass, basically biofilm formation. Sean did the C. elegans work, and Shane Christie, and a student in my lab did the biofilm work. All of these things in the middle are things that don't change the activity very much. Obviously, what we were looking for is something in this blue star, which was an optimized peptide that worked much better than either the 12 mer or the native 10 mer. Do those work in mouse models? This is again a oropharyngeal candidiasis model, and you can see the 12 mer works, 10 mer not so well, and then our lead compound at this point is what we call P4. P4 is the 10 mer sequence with a single amino acid change to it. Does that work in other formats? It does. This is a prophylactic model here as well, in which we simply mix the Candida inoculum and an intravenous infection model, and we look in the kidney and the liver 2 or 5 days later. Now, both of these, I think you can see the fungal burden in the kidney is significantly reduced with all of these different peptides, including our lead compound P4. Now, some of you will say, well, of course, you mix the inoculum with peptide, and the peptide kills the fungi, and so obviously it doesn't infect as well. Well, I'm going to tell you in a minute that this peptide doesn't kill fungi. It doesn't slow their growth. It doesn't have any effect like that. So this is a single treatment at the time of infection. Obviously, that's not the therapeutic model that you want to use. So we've tried a couple of other things. Now, the first one is to just see how many Candida albicans strains this works against. And here are 4 different strains compared to our type strain, SC5314, and it works for all of them. In this case, 3107 and 3184 are isolates from the same patient from an oropharyngeal infection. This was a young man who had HIV back in the early 90s and got oral candidiasis 16 times over an 18-month period. Treated with fluconazole, they eventually became resistant. And so here's an early isolate, and both P4 and fluconazole are effective against it. A later isolate, P4 is effective and fluconazole is not, as we would expect. We started playing, you may have seen data on the other slide, I wondered what that meant. We started playing with D-amino acid linkages. So this is P4 made with L-amino acids, and its half-life is not great. But made with D-amino acids, then its half-life exceeds 24 hours in human serum. P4D has all of the activities, and as we've optimized this, we've used P4L, P4D, 12 amino acid, whatever. But again, we've focused on this as now our lead compound that we're cleaning up some of these experiments on. It is active in oral models, in nematode models, in systemic models, and everything we've tested. This is a project in which we have on several occasions not done experiments because there's no way they're going to work. You heard from Flippos' talk that there aren't very many orally available peptides out there. This is orally available, and we were a little stunned by this. So this is a therapeutic model. We let the infection go for 2 days and then start giving the peptide either orally in drinking water or by IP infection. And as you could see here, both the oral dosing in the kidney and the liver not only works, but it works better than IP infections. That may be because of the dosing we gave them. We don't have any idea what the bioavailability is of this yet, but we're hoping that working with Dong and Juan here in the next few months, we can figure that out, where it goes in the body, what tissues it penetrates, and at what concentration. Histology, if you like that kind of thing, you can see here the black arrows, these huge masses of fungal growth this is the kidney, one here, one down here, one over here. And when you treat with P4D, there's almost none of that. So it really cleans the kidney out quite a bit. We also have just done our first Candida auris mouse model experiments. Now, Candida auris, as I said, is a big problem because of its drug resistance. And this wasn't the most beautiful data we had seen, but this is a tricky model because we have to immunosuppress the animals. But you can see here in the kidney, at least, there was a statistically significant reduction, again, in a therapeutic model when we let the infection go and treat several days later. So there's promise here, and we're really looking forward to exploring that more. Then I called up my friend Chung Yu Hung at UT San Antonio. She works on Coccidioides immitis. This causes valley fever, which is an endemic mycosis of the desert southwest of the United States into Mexico, Central America. And she uses mouse models, certainly, but she also just developed a Galeria melanella model. These are wax moth larvae. You inject the fungus into them and they die. Except when you then give them peptide, they don't die. The peptide was actually more effective at keeping Galeria alive than amphotericin B. Which is sort of the gold standard antifungal for this infection. Fungal burden was way down with amphotericin, although this didn't protect the animals. And it was modestly reduced here, but the animals live. So again, that's another disparate fungal pathogen that this might have efficacy against. So what do they do? Well, I'm going to tell you what they don't do first, what this peptide doesn't do. It's not toxic to red blood cells. It's not toxic to macrophages. It's not toxic to HeLa cells. And we've given a lot of mice this thing, and we've noticed no differences in behavior, weight gain, weight loss, any lifespan, anything else in these mice. So it doesn't appear, we have more toxicology to do, but it doesn't appear to be toxic under these circumstances. Somewhat to our surprise, it's also not toxic to Candida species either. As I said, we were using in mice 100 nanomolar. So this is 1,000 times higher, and it doesn't affect growth whatsoever. It also does not seem to synergize in vitro with a variety of stresses, including cell wall stresses, membrane stresses, temperature stresses, reactive oxygen species, antifungals, anything else. So whatever it's doing, it's not, the mechanism is not by killing Candida albicans. That might have some interesting properties because antivirulence agents, there's some thought there might be less pressure to develop resistance. Not sure I completely agree with that, but it is an interesting and atypical finding. So what does it do? What does it do? Giuseppe Buda-De-Societe and a postdoc in my lab started localizing INTV to the surface. This is the 68 mer version, but he has data that looks just the same with the shorter versions. And what you see here are spots in green of INTV localization, punctate, that appear and disappear. And Giuseppe, in a flash of brilliance or madness, one or the other, decided that this looked to him a little bit like what you would see when extracellular vesicles bud off of a Candida cell. And so we tested that. There's a dye that we just call DIL-C. It's in red in this image. And co-localization of that with a FITC localized, FITC labeled 12-mer is almost perfect, almost exact. So it does appear that the 12-mer is binding to places where extracellular vesicles are budding off of fungal cells. Why is this intriguing? Well, extracellular vesicles have been shown to affect host pathogen interactions, are required for biofilm matrix production, and a variety of other activities that are consistent with a significant role in virulence properties. He did this again with Cryptococcus gattii. Again, this is a very divergent fungus from Candida. And you again can see here the red of the DIL-C dye, the green of the 12-mer, and you see these co-localization spots, which we interpret to be extracellular vesicles that are caught in the cryptococcal polysaccharide capsule here. With, again, the help of David Andes, who has identified a number of mutants that produce fewer extracellular vesicles. Dave sent us quite a few of these, and I'm not going to show you all the data because it all kind of looks the same. This is one mutant, DOA4, which produces fewer EVs in vitro. This is the nematode C. elegans model again. If you infect with the wild-type Candida, they die. If you treat that here in the open circles with the 12-mer, they live. If you infect with the DOA EV deficient mutant, then they are not as virulent as the wild type, but they are also not as sensitive to the peptide. So that genetic data is consistent with a role in for EVs and inhibition of EV release by INTV as the mechanism of virulence inhibition. Dave then just simply tested, does this have any effect on EV release? And in the blue are the number of extracellular vesicles produced in a control treated cells versus the 12-mer, and you can see here there's about a 7 or 8-fold difference. So it really does look like somehow this short bacterial peptide is exerting an antivirulence effect on Candida through manipulation of extracellular vesicle quantity or quality in terms of the cargo that they might be carrying, which is a question we have not answered yet. So we never expected this project to get to where the point they are. We thought this would be a cool few experiments and then we'd move on. But we now have a lead compound derived from this bacterial protein that has potential for broad spectrum activity against a wide variety of fungal pathogens. We have shown so far that this an optimized peptide is stable, orally bioavailable, and effective against multiple pathogens. These interactions amongst the members of the microbiota, Candida with a variety of bacteria, are thus then an unappreciated source of potential future antimicrobial agents. And in fact, we've now identified that Candida is producing a small peptide that is active against Enterococcus faecalis. So this is a two-way war in this context. As I mentioned, we're hoping to get Dong and Huan soon to give us some help on PK/PD studies. We are extending our work here to other fungal pathogens through an application for an RFA that we just submitted, which include Aspergillus, coccidioides, the Mucormycota, Fusarium, and Candida auris. And we have a few more things to do to really confirm this EV mechanism of action. With that, let me just thank the people who did this work. First and foremost, Danielle Garson, shown here, the members of her lab, Melissa, and Sean in particular. Luis is really doing a lot of our animal work now. Giuseppe, Shane in my lab did a lot of, much of this work. It was started by Carrie Graham, and we have a variety of collaborators. And I will thank you for your attention.
Thank you, Phillips and Michael. Thank you very much for the great presentations. I guess we can go ahead to start the questions. If you have any question, please type in chat and we'll answer accordingly. So far, we have 3 questions. From the time, I guess those are questions for Phillips. And we're waiting other to type the question for Michael. So I will. Phillips, can you see the question? Maybe I also read the question for you to start. The first question from John is asking, is this considered uptake from stomach or small bowel?
Yeah, that's a good question. For the specific peptide, the 616, it is absorbed through the small bowel. There is precedence. Obviously, Novo Nordisk has claimed that their Rybelsus formulation is absorbed through the stomach, but the peptide that I was talking about is a small bowel, and most of the peptides we work in are on small bowel absorption.
Thank you. The second question is from Joshua, who's asking, could you give insight into formulations for delivering the permeation enhancer versus the peptide? For example, does the permeation enhancer need to be released first, or are they co-released into the intestine? I think that's a great question. I would like to ask.
Yeah, that's a great question. So the general belief is that the permeation enhancer needs to be co-released with a peptide. I mean, in the stomach or the intestine, depending on what you're targeting, right? That the action is driven by the simultaneous presence of the two. There's been a few studies many years ago, if you look at the literature, that actually, I don't recall the company that did the study, that they looked whether one could give the permeation enhancer ahead of the peptide and still get the effect. And while you will in theory think that might be possible, actually the exposures were lower when the permeation enhancer was giving ahead of the peptide. There's a lot of discussion in the field whether immediate release or enteric coating is needed. You can imagine that enteric coating will give you higher chances of this being together rather than immediate release that has to dilute in the stomach. But I was in a conference a couple of years back. Several people who participated, they basically said they don't routinely see this effect. So I think currently what I will say is you need to have them at the same time, generally speaking, but whether they need to be like, enterically or in the gastric environment, it's still being studied.
So
it's a very good question. I mean, yeah, it's a bit counterintuitive why it wouldn't work, because you will think you will first need to open the tight junctions and then the peptide gets absorbed. But I think it's just a spatial thing. I mean, the likelihood that these two molecules will end up at the same time, at the same place and everything. I mean, the gut is pretty non-homogeneous system.
I just want to add on to that question. So I forget if you mentioned in the presentation, but how much the permeation enhancer increase the bioavailability by what percentage? Do you have a rough idea, or that's commonly seen?
So it is very high depending on the peptide. For the one I
Peptides.
showed you, I mean, I showed you the data with and without Labrosol. If you go by AUC, it's the effect like 2 or 3 fold. So it goes from, let's say, 0.5 to 1% or something like that. If you look at some of the data on other peptides in the market, some of them go from 0 to something, so it's a much higher effect, and we've seen that also with some other internal work. So it is peptide dependent, so the system is not agnostic to the peptide properties. So you need to study this on the specific peptide that you're interested in. So I was interested, I mean, Michael, he mentioned that his peptide is orally bioavailable. It'd be interesting to see, how another peptide behaves at some point.
Yeah, I also feel interesting about the water intake you mentioned. Some other peptides seems have been greatly impacted by the water intake, which is very interesting. So I guess that's also peptide by peptide case, right?
Right.
That could be an interesting study, see what the peptides property can impact the water, impact to the bioavailability.
Yeah, the water effect, I mean, so the original paper that raised this question is, I believe, salmon calcitonin. If you look at the literature, there is an original paper with calcitonin that I don't recall what formulation they were using, but basically they showed a significant impact of water on that bioavailability. Now, their speculation, I mean, the authors of the paper speculated that it had to do with whatever formulation they were using. Again, I don't recall what it was stabilizing the peptide calcitonin against degradation. And by giving more water, you're basically diluting the formulation and it loses the stabilizing effect. So I think it's a reasonable hypothesis, but again, depends exactly what your formulation excipients are, whether this will be affected by this dilution effect or not.
Mm-hmm. All right, let's continue on this question on the panel. The next question is from Haiti. What is the expected recombinant peptide market in the future? That's the first question. Let's go with that question first.
I mean, there's several peptides. I mean, I don't know the entire peptide things out there. I mean, I don't. Maybe Michael has a better idea of what's out there on in his field. I mean, but on the oral side, I mean, we are working on 616 in phase 3. J&J has a peptide on IL-23 for they're currently doing psoriasis studies. So there are several companies that are looking at oral peptides right now.
So the next question is there a possible research opportunity in peptide postdoctoral? I guess that's a question regarding to career development. I don't quite understand this question. Maybe he's looking for like the postdoc at Merck to study peptides. I guess that's a question. Yeah.
I mean, it's a hot field. I mean, I don't have a postdoc position right now. I know maybe Dong or you, anyone else can comment on how much opportunities there are in the academia. I mean, I will assume there's a lot of interest in the field right now for postdoctoral research.
I think peptide is a very hot area right now, so definitely there should have some postdoctoral opportunity for that. All right, next question is for Michael. Is also from Joshua. Could you comment more on the activity of the D peptides? I would guess the molecular target is likely acarosine. I guess that's our question.
Yeah, so a couple things about that. There's a question 2 or 3 down from Diana Chow who asked about the mechanism of stability of the D-amino acid peptide. So it's thought that the D-amino acid versions are more resistant to serum proteases because of the chirality issue, they're not recognized as well. That clearly showed through in all of our versions of these peptides that are made with D amino acids, they're all more stable than the L amino acid versions. So that's consistent with what's known about the activity of these of the serum proteases. Now, what is a target? Is it achiral? Well, we don't really know. Our best data at this point points to it binding to mannan. So mannan is a carbohydrate. These are fungi. All of their extracellular proteins are O and N glycosylated with complex mannan chains. We have some biochemical evidence that it does bind to mannose residues, and there are some differences in the mannose side chains and the cell surface proteins in vesicles versus in just the plasma membrane as a whole. So if I were confident enough to in the mannan binding, I would have shown it to you in the presentation. So I would say that's preliminary at this point, but the carbohydrate will be achiral. The question is, those are very hydrophilic molecules, and our peptide is very hydrophobic, so that doesn't quite fit, which is one of my caveats to that data.
Thank you. So I guess you answered two questions. The next question from Song, which is my colleague. He's asking, are there any specific consideration to take into account when selecting permeating enhancer for peptide drug compared to small molecule drugs, Phillips?
Yeah, good question. So I don't think anyone knows yet the answer of, what exact permeation enhancer will work best for its peptide. I will say that in general, it doesn't appear that there is like, a magic bullet that, there is like some, permeation enhancer out there that, behave significantly differently than the others. Like if you look, most of the publications, whether they use like, caprate, SNAC, or some other fatty acid derivatives, they all give you this like roughly like 1 to 2%, bioavailability. Now, if you go to actual pharmacological agents, there's probably papers out there. If you look like, cholera toxin or specific inhibitors of tight junctions, you might see things, but okay, those are not easy to develop, right? There's other considerations with these things. Now, I personally, if you ask me, I don't think permeation enhancers are really like a thing that will significantly impact, most of the small molecule drugs. Most of the small molecule drugs have bioavailabilities 10, 20% at least. So adding 1% to a 10% bioavailability is noise. Right? It doesn't really add much. So I personally, I'm not really buying into using permeation enhancers for small molecule drugs.
Okay.
If I understood the question, if there's more clarification, please let me know.
Okay, there's one more question. Do you know or have plans to study the composition of the extracellular vesicles and how they benefit survival or okay, where is it? Okay, it just dropped something. The survival and or virulence of Canipaps. I guess that's a question for Michael.
Yeah, so there's a reasonable amount of data from Candida, from Cryptococcus, from a couple of other fungal pathogens that does not come from my lab, that is suggests that these vesicles are filled with enzymes that are important in cell wall remodeling, in biofilm matrix production. There are proteases and lipases in these vesicles that might mediate host pathogen interactions. Candida produces a significant number of extracellular proteases, and those target complement. They target other cytokines. They target human matrix proteins and things. So from that list, it is not hard to imagine that these are that the vesicle cargo is important in maintaining a fungal promoting host pathogen interaction. There was a paper not too long ago that suggested that these could work at a distance. And as you probably know, well, not as you may not know, the EVs are quite immunostimulatory. And for a lot of molecules, neutrophils, for instance, once they degranulate, if you can get them to do that at a distance, well away from the parent fungal cell, then they've kind of shot their wad with that. And that might be a way to reduce the overall antimicrobial activity of the innate immune system. Now, what is the peptide doing to that? Well, I showed you data that suggests that it's reducing the production of EVs. Is it also affecting the cargo of those EVs? That is a question we don't yet know the answer to, but yes, we are planning to test that. My guess is that it will, but that's why you have to do the experiment.
So, Michael, I have a follow-on question for you.
Yes.
Do you believe this extracellular vehicle belongs to like a host? Are you targeting the host? Because I remember NIAID, they try to avoid targeting the host, and they usually focus on targeting the pathogens. But do you see that will lead to that kind of the things later on that you need to? Do you think that the drug peptide actually is targeting the host itself?
I don't think that there's any therapeutic benefit that we can harness here, because as I said, the effects on the host seem to be immunostimulatory at a distance, by reducing or blunting the antimicrobial response, by breaking down the host extracellular matrix. The vesicle contents also promote the production of the biofilm matrix that help protect Candida from the cell. So I think our compound has therapeutic potential because it reduces all of those things. But figuring out a way to modulate the immune response to these vesicles is above my pay grade. You're going to need to find me somebody who's a much smarter immunologist than I am to do that.
Gotcha.
Kind of lost where we are. I guess this is a comment more than a question. The published Ribelsis study showed great specificity towards to peptide, water, etc. Semaglutide works, liraglutide does not, etc. So I guess that's not really a question. It's probably when we talk about earlier about the water. The next question from John is: any successful example of cell-permeating peptide as permeation enhancers?
So, I mean, if you have a cell-permeating peptide, you probably wouldn't need the permeation enhancer, right? The idea of those would be, they would get absorbed by themselves. I'm not aware of any specific cell-permeating peptide that's in clinical development right now, but I'm not tracking those as closely. I mean, there are obviously cell-permeating peptides that have been reported in the literature as tool compounds and so on. You will be looking at different pharmacology, obviously, because a non-cell permeating peptide will have different targets compared to a cell permeating peptide. So I mean, that would be something like specific to the interest there. But I'm not aware, but I don't track it that closely on what cell permeating peptides might be in development right now.
Okay, the next question from Dr. Dana Chow. The concentration dependent study showed 6 milligram had less effect than those with 12, 18, and 30 milligram, but not significantly difference between 12 to 30 milligram. What is the explanation?
Yeah, I mean, I guess every PK/PD relationship, at some point hits a plateau, right? As you're approaching the max effect, right? So this will indicate that the curve is somewhere between 6 and 12. Pick a number around 12, and then once you hit that number, the plateau is hit. And I don't recall on top of my head what is the EC50 or something, but I mean that would be a pretty typical PK/PD relationship. I'm sure Don or others can comment on that.
Okay, so the last one is also from John, but I guess it's still a comment said CPP might fluidize the membrane for target peptide to enter. So that's still considering the semi-permeating peptide. He was
Yeah, I
asking.
guess theoretically, was, I'm just not aware of any specific example that is looking at this effect right now in clinical development. It's possible there is. I just don't know what it is.
Okay. So that's all the question we have so far on the chat. Is anyone else have any question, or Dr. Liang, you want to summarize from today?
Well, yes, thank you. Thank you for the speakers to give us a very good insight, both from industrial perspective and from the actual clinical development of these areas of these peptides. A lot of these peptides, the studies we have seen it's very interesting and it's different. And we really appreciate both speakers' presentation and spending time to share your experience with us. And hopefully we can follow up more. And if anyone in the audience have more questions, we will collect it and maybe get in touch with the speakers. I guess we are about a few minutes to the time. I think at this point we can close off the meeting.
Thank you very much again.
Thank you.
Thank you all.
Thank
Thank all
you,
the audience to stay to listen to the great presentations. Really appreciate everyone. Thank you.
Everyone has a good spring break.
Take care. Thank you.